In this lecture segment, we will be discussing a structure within the kidney called the nephron. Now, nephrons are the structural and functional units that form filtrates. So these are the units that are going to be filtering the blood and processing it for wastes and nutrients. Nephrons are microscopic structures. We have approximately 1 million nephrons packed into each kidney. It's actually a little bit more than 1 million nephrons. There's about 1.2 million or so, but we'll just go with a nice round number at 1 million nephrons per kidney. So we've got a lot of these microscopic structures packed into each of our individual kidneys. Even though these are microscopic structures that we're talking about, the kidneys are still able to filter and process a very impressive amount of fluid per day. So the kidneys can filter a combined, or both kidneys can filter about 200 liters of fluid per day. So even though nephrons are very small structures, they're capable of doing this huge task of filtering and processing the blood for us. Nephrons come in two parts or two regions. We have the renal corpuscle and the renal tubule. So let's look at a diagram of what these nephron parts look like. On the left here, we have the renal corpuscle, and to the right, these tube-shaped structures here are going to be our renal tubules. Let's focus on the renal corpuscle here for a second, because the renal corpuscle has two parts to it. It has a structure called the glomerulus, which the glomerulus is a small cluster of capillaries. The glomerulus is where filtration is taking place. So we've got blood being delivered to the glomerulus from these afferent arterioles. Afferent arterioles are receiving their blood from the cortical radiate arteries that fan up and into the renal cortex. So blood passing through the cortical radiate arteries will be delivered to each afferent arteriole. The afferent arteriole will direct blood to the glomerulus. Blood will wind its way through the glomerulus, being filtered along the way, and remaining blood and fluid, so blood cells, proteins, and things like that, and any remaining fluid that's still in that blood will leave the glomerulus through the efferent arteriole here. The blood that is filtered while in the glomerulus is captured by a capsule or kind of a glove-shaped structure that fits around the glomerulus, and that glove-shaped structure is called the glomerular capsule. And so the glomerular capsule's job is to capture the filtrate that's leaving the glomerulus. The filtrate that's captured by the glomerular capsule is then directed into the renal tubules. The first region of the renal tubule that the filtrate will enter is this purple region here that you see on this diagram called the proximal convoluted tubule. The proximal convoluted tubule will direct that filtrate into a U-shaped region called the nephron loop. It's also called the loop of Henle. Finally, filtrate leaving the loop of Henle will enter the last part of the renal tubule that's called the distal convoluted tubule. That filtrate, after leaving the distal convoluted tubule, will be directed into a structure in teal here called the collecting duct here. The renal corpuscle is the place where filtration is occurring here. So blood is being filtered and the filtrate is being captured by that glomerular capsule. The renal tubules are where absorption of needed substances are happening. So the body is reabsorbing the water, the electrolytes, the glucose, and things like that from the blood that's in the filtrate, or from the filtrate, and putting it back into the blood. And the tubules are also working to secrete unneeded wastes into that filtrate. So the tubules are reabsorbing the things that our body needs, like water, electrolytes, and glucose that are present in that filtrate. And they are also secreting unneeded substances into the filtrate as well. The renal corpuscles and the renal tubules are located in slightly different regions within the kidney. The renal corpuscles are located within the renal cortex. The renal tubules begin in the renal cortex dip down or pass into the renal medulla and then return back to the cortex. So we see a couple of different regions of the kidney that these nephrons will pass through on their way through the body. So looking at this diagram here, slightly different diagram than the one I showed you previously, we see the renal corpuscle here, the glomerulus, which is that ball of capillaries that we discussed where filtration is happening, the glomerular capsule that surrounds it and is capturing that filtrate. So those two structures combined make up that renal corpuscle that we discussed. Filtrate that's captured by the glomerular capsule is directed into the renal tubules. That first region of the renal tubule is the proximal convoluted tubule. Proximal convoluted tubule then becomes that U-shaped nephron loop. Nephron loop then becomes the distal convoluted tubule and delivers that filtrate to the collecting ducts. Now, what I want to show you here is that this diagram is showing you the different regions that these structures are found in the kidney. So everything above this, this is actually the arcuate artery right here. So if we kind of label these here, this diagram does a poor job of labeling these arteries and veins here. So everything above this artery right here is this is the arcuate artery. Remember, the arcuate artery is essentially at the border between the renal cortex and the renal medulla, or the renal pyramids that are in the renal medulla. So this is the arcuate artery right here. So everything above this right here, including the renal corpuscles, is within the renal cortex. The nephron loops are the structures that dip down into the renal medulla. I mentioned the cortical radiate arteries that are feeding those afferent arterioles here. You can see the cortical radiate arteries here that are feeding the afferent arterioles that are going into those glomerular capillary beds there. And once again, just to remind you of what we're looking at here, or where we're looking for these structures here, is that the nephrons, with the exception of the nephron loops, are going to be housed in the renal cortex here, so that superficial layer of the kidney that we saw there. And we once again point out here the cortical radiate arteries that are feeding those glomerular capillaries, and the cortical radiated veins will actually be draining those nephrons and returning blood back to the systemic circulation. So let's look for a second here at the glomerulus. We'll kind of break down the parts of the kidney and discuss their function a little bit here. Let's begin with the kind of looking back at the renal corpuscles here. Let's look for a second at the glomerulus. Remember that the glomerulus is going to be a tuft or a ball of capillaries. It is a very well fenestrated set of capillaries. That means the fenestrations are small pores that are within those capillaries, and the endothelium that lines the walls of those capillaries has lots and lots of small pores in it, so that way fluid but not cells can pass from the glomerulus into the glomerular capsule. Just gave us another look at those fenestrated capillaries here. So remember, fenestrated capillaries have small pores in them. These small pores are large enough to allow fluids to leave the capillaries, but not blood cells or proteins. So in a healthy kidney, we'll see water in the filtrate, we'll see glucose in the filtrate, we'll see electrolytes like sodium and potassium in the filtrate. Glucose, water, and sodium and potassium will eventually be reclaimed by the renal tubules, but in a healthy kidney, we will not see red blood cells or proteins, because in a healthy kidney, these fenestrations or these pores in the capillaries are too small to allow the passage of blood cells or proteins. So the glomerulus is really where the filtrate is being produced. This is where blood is being filtered and essentially separated, right? We're separating some of that fluid of the blood that's going into the glomerular capsule. And the remainder of the blood, which is, again, fluid, but also cells and proteins, will leave the glomerulus and re-enter systemic circulation eventually. But the glomerulus's job is to produce filtrate. And filtrate is, as I mentioned before, it's a cell-free and a protein-free fluid. This filtrate will eventually become urine. So I think here it's important to make the distinction between filtrate and urine, because these are in fact two different substances. And I know in previous lectures I've probably been guilty of interchangeably using the two terms, but these two substances are in fact different from one another. There is a difference between filtrate and urine. Filtrate is the cell-free and protein-free fluid that is created at the glomerulus, and it comes from blood plasma. So that's what we had just discussed. Urine is the filtrate that has been processed heavily, that contains unneeded substances. So urine has been processed to reclaim a lot of water, a lot of the electrolytes, glucose that is found in filtrate. And it also contains a number of unneeded substances. So any excess salts like sodium and potassium will be released into the urine and ultimately leave the body. Metabolic nitrogen containing wastes like urea, that are the substances that are produced as a byproduct of protein breakdown. So those are the major differences between filtrate and urine. Urine is really just heavily processed filtrate that's going to be leaving the body. And we'll see this processing occurring later in the renal tubule. So we'll discuss the physiology of the kidney in a later lecture, and we'll look at how urine is produced. So continuing our discussion of the renal corpuscle, we'll continue to look at that structure by looking at the glomerular capsule. This capsule is also called Bowman's capsule, but I'll just refer to it as the glomerular capsule within my lectures. It is a cup-shaped structure, so it surrounds the glomerulus and collects the filtrate that is produced by those glomerular capillaries. The glomerular capsule has two layers. It has a layer that's essentially the outside of the capsule. It's called the parietal layer here. And that's this layer approximately right here. So it makes up the outside of the glomerular capsule. That is a simple squamous epithelium. So those cells are devoted to really just kind of preserving the external structure of the glomerular capsule. They have this parietal layer. These cells in that parietal layer have no contact at all, or do not directly touch the glomerular capillaries in any way. The deep layer here, if we zoom in on this diagram here, the deep layer here, called the visceral layer, actually does make contact with those glomerular capillaries. And the cell type that we see here is a different cell type from the parietal layer. The parietal layer had a simple squamous epithelium. The visceral layer has the special cell type called podocytes. This term podocyte literally just means foot cell. And these podocytes have little extensions, feet-like processes on them that make contact with with those glomerular capillaries there. Getting even a closer look at the junction between the glomerular capsule and the glomerular capillaries, we see three structures. The fenestrated epithelium, or endothelium as it's called here, that is the epithelium that makes up the wall of the glomerular capillaries here. There is a basement membrane made out of sugar proteins, glycoproteins. It's essentially a glycoprotein glue that holds the podocytes or glues the podocytes to the epithelium of the glomerular capillaries here. And that layer is called the basement membrane. So filtrate that's leaving the glomerular capillaries will exit through the pores here, move directly through this basement membrane. Even though this basement membrane is a solid structure, it really doesn't do much to impede or inhibit the movement of fluid through it. So the filtrate is squeezed through those fenestrations, squeezed through the basement membrane, flows between the podocytes and into the glomerular capsule here. We take a kind of a different view of what that looks like here. We see that there are these little slits called filtration slits that exist between the foot processes of those podocytes here. These blue arrows represent the direction of filtrate flow here. So this would be filtrate being squeezed by the pressure of the blood that's in the glomerular capillaries between the fenestrations here, that is between those pores in the glomerular capillaries. That filtrate will be also squeezed through this basement membrane, which looks solid, but again, like I said, does not really do much to impede the movement of fluid through it. And also finally, between these foot processes and into the glomerular capsule itself. So these foot processes, these filtration slits, allow fluid to move into the glomerular capsule, but not cells or proteins. So these filtration slits and fenestrations prevent cells or proteins from getting into the filtrate of a healthy kidney. If we look at a scanning electron micrograph of what these podocytes look like, you can see the cell body of a podocyte here. What you're not seeing underneath it is the glomerular capillary underneath this podocyte here. The podocytes wrap them so tightly that we can't see it in this image here. But what you are seeing here are these little foot-like extensions of the podocytes here. And in between them, little gaps, these little dark gaps that are between them are the filtration slits that we just talked about. Now, to me, the podocytes actually look a little bit more like feathers. I probably would have named them feather cells instead of foot cells, but I didn't get to name them, so there you have it. I wanted to highlight this structure here since we've already talked about it in a couple of contexts. The structure called the filtration membrane. And the filtration membrane is made of these three layers that we talked about previously. The capillary endothelium, which is the wall of our glomerular capillaries. The basement membrane, which is the glycoprotein or sugar protein glue that fuses the capillary endothelium to the foot processes of the podocytes in that glomerular capsule there. So the filtration membrane consists of these three structures: the blood vessel epithelium of those, or these cells that make up the wall of those glomerular capillaries, the podocytes and their foot processes of the glomerular capsule, and the basement membrane that fuses them all together. So blood from those cortical radiate arteries will enter the afferent arteriole here. It will circulate through the glomerulus here. This is just another diagram of a glomerulus, so different perspective of it, but same structure that we saw previously here. Blood will enter these afferent arterioles, wind its way through these glomerular capillaries, and then exit the glomerulus through the efferent arterioles here. Blood pressure in this bed of glomerular capillaries pushes some of that blood plasma, which is our filtrate, out and into this glomerular capsule here. It's called the Bowman's capsule, so that's the other name for it. So this glomerular capsule is capturing some of that filtrate and directing it into the proximal convoluted tubule here. So we've looked in detail at the renal corpuscle. We'll turn our attention here to the renal tubule, which will be receiving filtrate from the renal corpuscle and processing it. So the role of the renal tubule is to produce urine. And in order to do that, the renal tubule is going to have to do two things. It's going to have to reabsorb water, electrolytes, and glucose from the filtrate because we need that stuff. And so we don't want all of that to be unnecessarily lost to the urine. So the renal tubule is going to be reabsorbing the substances that our body needs. And it's also going to be secreting unneeded wastes into that filtrate as well. So the job of the renal tubule will be to absorb and secrete. So let's take a look again at the renal tubule here. The renal tubule, now this diagram doesn't really give a good approximation of its length. The renal tubule in a living kidney is a little over 1 inch long. And we saw the three parts of it earlier, but we'll review those again here. Those three parts are the proximal convoluted tubule, which receives filtrate from our glomerular capsule here. Filtrate will then be directed into the nephron loop, which is also known as the loop of Henle. Filtrate then flowing through the loop of Henle, then arrives at the distal convoluted tubule, and the distal convoluted tubule will be the last segment of our renal tubule. Each of these three regions of the renal tubule consists of a single layer of epithelial cells, and each of these regions has a specific epithelial cell type and function associated with it. So if we look at the proximal convoluted tubule, we see that the epithelium of the proximal convoluted tubule is a simple cuboidal epithelium. So we have these nice cube-shaped cells. This surface up here is the apical surface. So this is the surface across which filtrate will be flowing. So if we removed these labels here and we looked at it in a real living kidney, we would see that filtrate would be proceeding along through the proximal convoluted tubule up here. The structure that I want to point out here that's going to be really helpful in absorption are these apical microvilli here. This is also called the brush border here. So we also saw a brush border in the small intestine that were made of microvilli. The microvilli go by the same name here as they did in the small intestine. And here they're also performing the same task. They're increasing the surface area. So these microvilli grant these cells a huge amount of surface area. So these guys are able to function really well in reabsorbing water, electrolytes, and glucose from the filtrate. We also see lots of mitochondria in this because this process of reabsorbing and secreting is going to be a very energetically expensive process. And so we need lots of mitochondria to make ATP to fuel that process. So the proximal convoluted tubule will be reabsorbing water and solutes for us and also secreting unneeded wastes into the filtrate as well. And so for both of these, we see that we have a lot of mitochondria and a lot of cellular energy that's going to need to be created in order to fuel that process. Turning our attention to the nephron loop, or the loop of Henle here, we described it previously as a U-shaped structure with two regions to it, or two limbs. They call them limbs in this diagram here. These limbs are referred to as either thick or thin segments. You can see they kind of change in thickness in this diagram here. They're not labeled as thick or thin segments in this diagram, but in other diagrams you'll see that they're labeled as thick or thin segments. The nephron loop has thick and thin segments because the thick and thin segments reflects the type of epithelium that is there. The thick segments have a cuboidal or a columnar shaped epithelium, so these are really tall epithelial cells. The thin segments have a flatter squamous epithelium, so those are those flattened epithelial cell types that we've discussed before. So if we look at the thin segments, we see that the thin segments are made of a flattened squamous epithelium. They have some microvilli here, but not a whole lot. So we have some absorption and secretion taking place in the nephron loop, but a lot of that absorption and secretion has already taken place in the proximal convoluted tubule, so we don't need quite as much of that here. Finally, the distal convoluted tubule, which is the last segment of our renal tubule, has a simple cuboidal epithelium, so it's a taller epithelium type, just like we saw in the proximal convoluted tubule here. These cells have very few microvilli, so if we turn our attention to the cells of the distal convoluted tubule, we see that they are a cuboidal epithelium, single layer, just like we've seen in all the other regions of the renal tubule. We don't see as many microvilli on their apical surface, so these guys are mostly functioning in secretion rather than absorption. So the job of the distal convoluted tubule is primarily to add wastes to the filtrate rather than absorbing things. A lot of the absorption has already taken place in the proximal convoluted tubule. Although this is not part of the nephron drains into these straight collecting ducts here. The collecting ducts all end in a bundle at the papilla of the renal pyramid. So what comes out of the end of the collecting ducts is urine, and that urine will be directed to the ureters, as we saw in previous lecture segments here. The collecting ducts have two cell types. They have principal cells and intercalated cells. The principal cells are the most common cell type in the collecting duct. We see that they are cuboidal in shape, have very few and very short microvilli, so the microvilli that are present are very short. The principal cells, their job is to maintain water and sodium balance in the body. The intercalated cells are also cuboidal shaped, just like the principal cells. We see that they have many more microvilli, and those microvilli are much taller. Their job is to maintain acid-base balance within the blood. So as I mentioned, the collecting duct receives filtrate from many nephrons. So in this diagram here, we just see one nephron, but these little stems that you see here that are essentially cut at the collecting ducts here, these are the ends of the distal convoluted tubules of many nephrons. So you can see that many distal convoluted tubules will be directing their filtrate into these collecting ducts. And all of these collecting ducts, as I mentioned, are located in or are bundled together and end or terminate at the papilla of the renal pyramids. These ducts, the ducts themselves, the collecting ducts themselves, are actually located in parallel to one another inside of the renal pyramids. These are what give the pyramids kind of a striped appearance, because all of these collecting ducts are oriented parallel to one another. So because of that parallel orientation, the renal pyramids have a sort of striped appearance to them. All of those collecting ducts terminate at the papilla of the renal pyramids here, and so the substances that will be coming out of those collecting ducts is urine, and that urine will be directed into the ureter towards the urinary bladder. So we've looked at the basic structure of the nephron and the collecting ducts. Let's return for a second back to the nephron and look at types of nephrons. So we actually have two different types or varieties of nephrons within our kidney. The first that we're going to take a look at here is called the cortical nephron. You can probably guess where they're located. These guys are almost entirely confined to the renal cortex. The exception here is that the nephron loops of the cortical nephrons dip a little bit into the renal medulla. But for the most part, the cortical nephrons are located within the renal cortex. They make up the vast majority of the nephrons in the kidney, so they make up about 85% or so of the population of nephrons that's in the kidney. They're characterized by very short nephron loops. The remaining 15% or so of the nephrons are a type of nephron called the juxtamedullary nephron. They are really kind of located at the junction between the renal cortex and the renal medulla. So they really kind of sit at the junction between the renal cortex and the renal pyramids. They're characterized by very long nephron loops. The reason they have those extremely long nephron loops is because these guys are important for producing highly concentrated urine. So these are the nephrons that go to work when we're dehydrated to conserve water. So we don't have as many of these, we don't need as many of these within the kidney, but they do serve a very important function. This diagram just shows the two different types of nephrons here. So on the left here, we have a cortical nephron with its short nephron loop. On the right side here, we have a juxtamedullary nephron with this extremely long nephron loop and its associated blood vessels. So let's spend a minute and actually look at these blood vessels that are associated with the renal tubules. We've got two different types of blood vessels, or I should say two different types of blood vessels depending on the nephron type. So cortical nephrons have capillaries that are associated with them called peritubular capillaries. In juxtamedullary nephrons, those blood vessels are called VASA recta. So let's spend a minute and look at those nephron capillaries. Cortical nephrons, as I mentioned, have peritubular capillaries that surround the renal tubules. Juxtamedullary nephrons have what are called vasa recta that surround their renal tubules. And the vasa recta in the juxtamedullary nephrons are mostly surrounding the nephron loops. Both of them arise or come from the same structure. They actually come from those efferent arterioles. And so if we go back and take a look at this diagram here, we see that, just once again to give you an orientation here, these are this would be the arcuate artery right here. So this is at the junction between the renal cortex and the pyramids in the renal medulla. Blood flow or fluid flow is indicated by the arrows here. So blood would be flowing up from the arcuate arteries into the cortical radiate artery here. These cortical radiate arteries fan up and into the renal cortex here. From the cortical radiate artery, we get little branches off of it that are the afferent arterioles that serve each of the glomerular capillaries here. So whether it's a cortical nephron or a juxtamedullary nephron, it makes no difference. We still have the cortical radiate arteries branching off and delivering blood to the glomerulus of each of the different nephron types here. Filtration we saw happens at the glomerulus and any remaining fluid and blood cells, or I should say any remaining fluid and all of the blood cells and proteins that are leaving the glomerulus leave through the efferent arteriole, and then the efferent arteriole very quickly becomes, in cortical nephrons, very, very quickly becomes the peritubular capillaries that are in close association with mostly the proximal convoluted tubule and a little bit of the distal convoluted tubule as well. So the peritubular capillaries are surrounding the proximal and distal convoluted tubules of those cortical nephrons. And eventually those peritubular capillaries drain into the cortical radiate veins and leave the renal cortex there. Same thing that we see, or I should say same configuration here in the juxtamedullary nephrons, where we've got the afferent arteriole feeding the glomerulus here. Blood leaving the glomerulus is then directed into the vasa recta, which surround the nephron loop of the juxtamedullary nephrons. And then blood is eventually returned from the vasa recta back to those cortical radiate veins there. So turning our attention to peritubular capillaries, we see that the peritubular capillaries are very porous, so they're functioning in absorbing both water and solutes from the fluid surrounding the renal tubules there. So their job is to pick up what the renal tubules are absorbing, and carry that back to systemic circulation. They empty their contents into the cortical radiate veins, as we talked about in the previous slide there. The vasa recta are the blood vessels that are associated with the nephron loops of the juxtamedullary nephrons, and they have the same configuration. So they're functioning to absorb water and solutes from the space surrounding the from the space surrounding the nephron loops, and carry that material back to the blood. Their function is to form highly concentrated urine, so we'll see that come into play when we start talking about kidney physiology later. So let's also turn our attention just briefly to a structure that's in close proximity to our renal corpuscle here, and it's also part of the nephron as well, since it represents the coming together of the ascending loop, or the passing in close proximity of the ascending limb of the nephron loop to the renal corpuscle here. And this structure is collectively called the juxtaglomerular complex, or JGC here. The juxtaglomerular complex is actually part of the nephron loop, the ascending limb of the nephron loop, on its way to the distal convoluted tubule. So this ascending limb passes in close proximity to the afferent and efferent arterioles that are serving the glomerular capillaries here. The juxtaglomerular complex has a couple of cell types in it that characterize it. They have a small population of cells called the macula densa cells that are actually within the ascending limb of that nephron loop there. The mesangial cells, which are kind of situated between the ascending limb of the nephron loop and the afferent efferent arterioles, and these granular cells that surround the superficial surface of the afferent arterioles. So each nephron has a single juxtaglomerular complex. The function of the juxtaglomerular complex is to regulate blood pressure into the glomerulus, that is, blood pressure getting to the glomerulus, and it also regulates the rate of filtrate formation. We turn our attention to the juxtaglomerular cell types. We'll return to those macula densa cells. These are sodium chloride detectors, so these guys are measuring sodium chloride concentrations within the filtrate. So we can see that filtrate, or from this diagram here, we can see that this is where filtrate would be passing through. This is the lumen of the ascending limb of the nephron loop here. The macula densa cells are situated in a place that they're able to sense the amount of sodium and chloride that's within the filtrate as it passes by those cells there. The granular cells are a population of enlarged smooth muscle cells. And they're situated between the ascending limb of the nephron loop and the afferent and efferent arterioles that are serving the glomerulus. So these are enlarged smooth muscle cells. These are blood pressure detecting cells. And they also store an enzyme called renin, and renin is important in helping to control the rate of filtrate production in nephrons, and it also controls blood pressure as well.